In this section, we will examine knowledge distillation through the microscope of nDNA. Knowledge distillation is a widely adopted technique for compressing large language models, LLMs, by training a smaller, more efficient student model to mimic the behavior of a larger teacher model. Introduced by Hinton and colleagues in 2015, this method aims to preserve performance while reducing computational overhead, enabling deployment on resource-constrained devices. Over time, the field has evolved to include techniques like intermediate representation alignment, feature transfer, and task-adaptive knowledge distillation. Traditional evaluations of distillation have focused largely on surface-level metrics such as accuracy, loss, or perplexity without interrogating its effects on the model's internal epistemic geometry. In this work, we reframe knowledge distillation through the lens of neural genomics, treating the teacher's latent structure as a form of epistemic genome and the distillation process as a form of latent genome compression. Let's understand the distillation setup, what we did and why it matters. To see how distillation reshapes a model's internal geometry, we ran controlled teacher-student experiments and read them through nDNA. Our student was Llama 38B. Each teacher was a culturally fine-tuned variant, Africa, Asia, China, Latin America, the Middle East, and others, so every distilled offspring inherited a distinct cultural prior from its teacher. Pipeline. Students were trained with classic soft-label matching on teacher logits. The evaluation prompts were drawn from civic, moral reasoning, civic values, legal norms, so we could test factual reproduction and conceptual generalization. What nDNA measures? Instead of judging at accuracy, we trace the latent manifold across layers using three geometry signals: spectral curvature, how sharply representations bend; thermodynamic length, how much semantic effort the path expends; and torsion, how much the path twists out of plane. From these we derive simple, layer-ways readouts, inheritance deviation, curvature gap, and length gap to see where the student truly follows the teacher's backbone, where it compresses without loss, and where it flattens, drifts, or overbends. Objective. Our goal is to understand structural variations or health in the student's nDNA. Does curvature collapse? Does length contract? Does torsional diversity disappear? In aggregate, as visualized later, we find that distilled students tend to settle into a compressed geometric basin, flatter curvature, shorter length, and muted torsion, highlighting the latent genome costs of compression even when surface accuracy looks fine. This setup lets us compare teachers, quantify epistemic retention, and decide where to add bridges, adjust temperatures, or reweight intermediate losses to preserve the beliefs we actually care about. Let's see this in action. What we learn is distillation trains a smaller student to copy a teacher. The student model is usually the teacher's soft logits with a temperature, sometimes plus intermediate losses that nudge internal states. In nDNA, students tend to flatten, spectral curvature drops, thermodynamic length shortens, and belief field strength weakens. The model keeps the right answers but with a compressed manifold, less bend, less effort, less directional steering, so it can be less robust of distribution. All the students' models collapses toward a compressed basin in latent space. In short, phenotype preserved, morphology simplified. Distillation is unidirectional imitation. The student often inherits the teacher's behavior while diverging in anatomy. Internal geometry is simplified even when accuracy holds. That's why students can pass the test, yet show reduced plasticity, weaker torsion steering, and poorer robustness under distribution shift. Merging in contrast to trained parents are recombined layer by layer. High salience directions are preserved, low salience ones interpolate. In nDNA, this shows a recombination bulge in the middle layers, kappa and length rise, followed by a settled backbone in later layers, sometimes dominated by one parent, sometimes balanced, and sometimes emergent in regions neither parent used. Rather than compressing, merging tends to restructure or enrich the manifold, maintaining bend, effort, and directional belief, traits that often translate into stronger generalization and more stable behavior under shift. In the plot, dashed purple trajectories are the merged offspring, ethers, solid colored curves are the cultural parents, and the black dotted cloud marks the distilled students. Bottom line. Distillation is equal to efficient imitation with compression. Merging is equal to capability recombination with structure. Choose distillation when footprint rules the day, but guard against latent collapse. Choose merging when you want blended or novel capabilities and can manage mid-layer recombination. Let's discuss the epistemic consequences of knowledge distillation. Distillation is often praised for preserving behavior while cutting size, but the nDNA lens shows a hidden cost. It reshapes the geometry of reasoning. A teacher's internal, semantic genome has 3 hallmarks measured layer by layer. 1. Spectral curvature, 2. Thermodynamic length, and 3. Belief field strength. In practice, distillation maps this rich set to a variance, collapsed manifold, curvatures flatten, lengths shorten, and belief forces weaken. This is a genetic bottleneck in latent space. Why does this happen? In information geometry terms, distillation tends to shrink the teacher's Fisher metric and the curvature spectrum. Think, the spread of principal bends. The student's Jacobian, its map from inputs to hidden states, often has a narrower singular value spectrum, so exploration collapses toward a central basin. You keep the phenotype, the answers, while simplifying the morphology, the manifold that yields those answers. Bottom line, distillation often preserves the answers while thinning the mind's geometry. Intuitively, picture reasoning as a path. Curvature is the model's ability to turn toward evidence, length is the work it's willing to do, and belief strength is the compass that keeps it oriented. If the simple scaffold score, curvature times length times belief strength, slides downward as layers depend, the student becomes flatter, lazier, and less guided. It can pass tests yet wobble off distribution. In practice, that means more brittle behavior, higher guardrail costs, and less trust in new contexts. Please refer to the book for further details. Let's have genomic analogy on knowledge distillation. To understand, we have to revisit 3 foundational concepts in genomics. First, bottleneck effect.
Do you know what the bottleneck effect is? Let me explain. The bottleneck effect occurs when a population undergoes a drastic reduction in size, often due to environmental factors such as natural disasters, habitat loss, or human activities. During a bottleneck event, the genetic diversity of the population is dramatically reduced as only a small subset of individuals survive to reproduce. As a result, certain alleles or gene variants may be lost entirely from the population, while others become more prevalent due to genetic drift. The consequences of the bottleneck effect can be far-reaching, impacting the population's ability to adapt to future environmental changes and increasing its susceptibility to genetic disorders. In conservation biology, understanding and mitigating the effects of bottlenecks is crucial for the management and preservation of endangered species.
Second, epigenetics.
The sweet smell of fruit doesn't normally send rats running, but when researchers paired the orange, cherry, almondy scent of the chemical acetophenone with a painful electric shock, lab rats quickly learned to fear it. Along the way, extra neurons sprouted in their noses and in the smell processing center of their brains, making them super sensitive to the scent. This result isn't shocking. What is surprising is that the rats' pups and their pups' pups were also startled by the smell of acetophenone and had the same extra neurons as their fathers, despite never having been introduced to either their dads or the fruity scent before. But how could the pups have inherited something that their fathers learned? Basic genetics tells us that only DNA gets passed along to offspring. Characteristics like memories, scars, or giant muscles can't get passed on since acquiring them doesn't alter the genetic code. But it turns out that instilling fear in the rats did trigger genetic changes, not in the DNA sequence itself, but instead in how that code was read and used in the rats' bodies. In every cell, biological machinery constantly translates DNA into the proteins needed to carry out vital processes. Chemical switches attached to the DNA turn genes on or off or up and down, telling the machinery which proteins to produce and in what quantities. These switches, called epigenetic tags, are why a kidney cell looks and acts differently than a skin or nerve cell, even though all 3 cells have identical DNA. But the switches in any one cell aren't set in stone. Teaching those rats to fear the fruity smell switched one of their smell-sensing genes into overdrive. Researchers don't know all the places in the rats' bodies where this switch got flipped, but they know it happened in one key set of cells, the rat's sperm cells, which would one day pass along the tweaked genetic material, making the next generation of rats super sensitive to acetophenone. Rodents aren't the only creatures demonstrating this weird type of inheritance. In Uvakalik, Sweden, boys who suffered through tough winter famines went on to have super healthy sons, with extremely low rates of heart disease and diabetes. And their sons had the same excellent health, living an unbelievable 32 years longer on average than the grandsons of boys who hadn't gone hungry. To be clear, this does not mean that we should start starving our kids for the benefit of future generations. Scientists don't even know yet exactly which switches the Swedish famines flipped. While we have been able to connect specific epigenetic changes to health effects in mice, we're a long way off from being able to make those connections in humans. That may sound like a bummer, but it's mostly because we humans don't live in the well-controlled environment of a laboratory. And for that, we should be grateful.
And the third, Hardy-Weinberg equilibrium.
One of my favorite topics that involves biology and math and is also absolutely fascinating is the Hardy-Weinberg equilibrium. It got its name from a mathematician and a physician, and it states that a population's allele and genotype frequencies are constant, unless there is some type of evolutionary force acting upon them. A reminder from our ecology videos, a population in this case is a group of organisms that are all the same species and can breed with each other and have fertile offspring. However, there can be variety among a population as they are not clones of each other. But based on Hardy-Weinberg equilibrium, the population's allele and genotype frequencies would remain constant if no evolutionary force acts upon them.
Think of the teacher as a big population of reasoning modes and the student as a tiny descendant formed through a narrow channel. First comes the bottleneck effect. Because the teacher-to-student path uses one parent, a fixed temperature, and a limited prompt set, the effective population size is small. In genetics, that means diversity drops quickly. Here it means only a narrow slice of the teacher's reasoning modes gets through. Next comes Hardy-Weinberg disequilibrium. The usual assumptions, large population, no selection, random mating, are all broken. The loss pushes the student in one direction, that's directional selection to match logits. The small channel introduces drift, and together they reduce diversity. A simple way to say this is with expected heterozygosity. H equals 1 minus the sum of each mode's share squared. Under distillation, H falls as a few modes dominate. If you train several students from the same teacher, FST, the between population differentiation, tends towards 0, the students become homogenized copies even if their test scores look fine. Finally, epigenetic inheritance. What actually crosses the channel are regulatory marks, soft labels, temperatures, and intermediate hints, not a full copy of the teacher's parameters. Those marks silence some pathways and promote others, yielding canalization, stable, stereotyped responses with less internal variety. The nDNA view lets us see this inside the model. Layer by layer, we track 3 signals. 1. Spectral curvature. 2. Thermodynamic length. And 3. Belief field strength. Under distillation, curvature flattens, length shortens, and belief strength shrinks into a narrow cone. The model keeps the teacher's answers, but its internal manifold is simpler and less adaptable. A practical single gauge is a scaffold score, curvature times length times belief strength, read at each layer. If that score steadily drops as depth increases, the student is losing the ability to turn, to think longer, and to stay oriented, so it will sound right on familiar data yet wobble when the distribution shifts. In short, phenotype preserved, morphology simplified. Please refer to the book and the website for details.